Welcome to the channel. I hope you all have watched my previous video on enzymes. After studying that, did you try to think how does a cell know when to increase the enzymatic reactions, when to slow down the enzymatic reactions? What is the exact mechanism behind this control? It's really what we are going to uncover from the video today. Think of an ongoing game in a ground or in a court, maybe a basketball match, volleyball match, cricket match, a rugby match, or anything. In a game, always the players try to uh, pay their attention, they make their complete effort to have a attractive scoreboard. What about the audience who are watching the game? What do they expect? Except the victory of their corresponding team. They need to have a good time. They need unforgettable memories. Simply, they need entertainment. So, from what are they got entertained? It's not only the score, the way how the scoreboard changes, the way how the players move in the ground or court, how they tactically position in the court, those things will delight the crowd. So I'll narrow down this scope to a cricket match. Uh, Assume that you are in a cricket match among the audience, and suddenly the coach of the team comes with a new game plan and sends a message to the two batsmen in the ground. What's that? Increase the batting speed. We need 100 runs in the next 10 overs. Now what will happen? Now the audience get a chance to see a good game. Aggressive hits over boundaries, good run in between wickets. Not only that, fantastic deliveries and brilliant catches, enormous fielding efforts. They would delight the crowd. Think of the opposite. Now the situation is changed. Now the coach needs to stop the falling of wickets. So this time he asks the batsman, don't try to score, just try to keep the wicket. We just need to escape the match loss. Two different situations. So in this example, the ground is like the cell, and the effort, the strategies sent by the players, they are like the metabolic reactions within the cell. And the wickets, the score, they are like the metabolic products. So, who are the players? Players are like the enzymes. And the coach, who is he? The coach is like the enzyme regulatory molecules. That's the topic, enzyme regulation. Regulatory molecules of enzymes. Most of the enzymes need regulatory molecules. Out of them, allosteric regulation is one type. Let's see what's this allosteric regulatory molecules. If we consider an enzyme with several subunits, think this is the enzyme. Now you can see it has 3 subunits. Now they are subunits. Each subunit has an active site. They are the active sites. So, other than those active sites, there are some special sites. We call them allosteric sites. We call so because they are the exact places where the allosteric regulatory molecules get attached to the enzyme. If this is the enzyme, this is the allosteric site. Don't get complicated. They are the active sites and it is the allosteric site. So, to make the function of the enzyme, it needs the help of the allosteric molecules. You can see now allosteric molecule has attached to the enzyme. Assume now this is the enzyme that is activated by allosteric molecules. So here to the allosteric site activator has bind. This is an activator. When an activator is bound to the enzyme can get attached with the substrate molecules than previous. In other words, the enzyme always stabilizes the active form. This is the active form because the active sites have the ability to attach with substrate molecules. And this is called allosteric activation. And if the activator releases from the enzyme molecule and inhibitor gets bind. Now instead of activator there is a inhibitor. Now you can see the active sites have changed their shape. Now the substrate molecules cannot get bound with those active sites. What is the reason for this change? It is the binding of this inhibitor, and this is reversible. This shape we call this the inactive form always when the inhibitor releases from the enzyme and attach with the activator, it stabilizes the shape of the active form. This is what we call allosteric inhibition and allosteric activation. It is one type of mechanism in the enzymes. Now you know the negative effect and the positive effect of the allosteric molecules. The negative effect is the allosteric inhibition. The positive effect is the allosteric activation. There is another type. It is called feedback inhibition. What's this feedback inhibition? Feedback inhibition is where the enzyme always controlled by the end product. Here, at the beginning, the substrate gets bind to the enzyme and make the enzyme substrate complex and turn into final products. The final product get binds to a early enzyme of the metabolic process. So as a result of that, the shape of the enzyme is changed. When the shape is changed, no more substrates can get bind to the enzyme. Therefore, the further production of the certain molecule is stopped. We call this feedback inhibition. Simply, this is like a self-control. In other words, the end products themselves go to the enzyme and says, it's enough, we have already produced enough. This is important for the metabolic functions within the cell, because to reduce the wastage of the resources, to reduce the overproduction, this feedback inhibition is important. This is like if I explain this further, this is like a tank in which the inlet is controlled by a floating valve. When the water flows, when the tank fills, the valve closes the inlet. When the water level goes down, it fills again, a self-control. So the feedback inhibition is a allosteric regulation found in the cells. Three mechanisms regarding the allosteric regulation: allosteric inhibition, allosteric activation, and feedback inhibition. There's one more. According to your syllabus, it's called cooperativity. Let's see what cooperativity is. Cooperativity is when an enzyme gets bind with the substrate molecule, all the other active sites also get stimulated. Assume the enzyme is like that, and they are the active sites. There are a large number of active sites. That's why you need a small concentration from enzymes. I described them in the previous video. So they are the active sites. Now assume this is a substrate molecule. And here the first substrate molecule attached when the first substrate attached to an active site of the enzyme, all the other active sites also get stimulated. That means the affinity of the other active sites also increase. This is like pulling out of a biscuit from a biscuit packet. It's bit hard to pull the first biscuit. Thereafter, it's very easy. So the cooperativity is also like that. After the first substrate molecule binds to the enzyme, then it becomes a teamwork, and likewise they increase the metabolic reaction. The first substrate binds, then there is a slight change in the other active sites. Thereafter, it's very easy for the substrate molecules to bind, and then there is a rapid increase in the enzymatic reaction. This is also the real reason for having the sigmoidal shape in the rate of enzymes and the time. So that's about the allosteric regulation. It is a powerful control. Both the feedback inhibition and cooperativity, they are powerful control mechanisms with relevant to the allosteric regulation. So let's meet again in our next video. Thank you for watching.